Well, wake up, folks, because like they said yesterday, we have a long place to go and a very short time to get there. And we're going to talk about angiogenesis. I want to just say that my original talk was a lot longer. I had to cut it down for this. But the person who got me interested in this field of angiogenesis happens to be sitting right there. And he did a very interesting experiment many, many years ago. And that's using a fertilized chicken egg, put a drop of octreotide on a piece of filter paper, and then put that on a fertilized chicken egg, and showed that it inhibited angiogenesis in that chicken egg. And that led him to do 6 chicken eggs. He then taught me about this, and I only did 24,000. But anyhow, where are we today in angiogenesis? Well, first, what's angiogenesis? New blood vessel growth. Is it just cancer? No. The 3 major causes of blindness, all angiogenesis driven. Psoriasis, angiogenesis driven. Rheumatoid arthritis, the joint destruction, angiogenesis driven. And the idea of controlling angiogenesis in cancer is that you will never cure it, but you will make cancer a chronic disease. You will die with the disease rather than die of the disease. Well, what are the big problems in developing effective anti-angiogenic agents? Well, number one, with all respect to Dr. Shaw, people use either animal models or they use cultured human cells. The trouble is that cells need to talk to their neighbors, and their neighbors aren't like them. In other words, if you have a breast cancer, that breast cancer is built of breast cancer cells, nerve cells, blood vessels, and a whole set of cells called stromal or supporting cells, and they form a critical structure. And without that structure, the tumor can't grow, the blood vessels can't grow. And current assays are, I think, in many ways non-specific. So my laboratory developed what is called the human angiogenesis model. And the human angiogenesis model is really pretty nifty. Basically, we take either a piece of blood vessel from a placenta, or we take your tumor at the time of surgery and take a very small chunk of it, 1 millimeter fragment, and as you think about a tumor, it has millions of blood vessels in it. So when I cut across your tumor and make it into a 1 millimeter cube, there are bazillions of blood vessels that are right beneath the surface. And if I can encourage them to grow, they grow out into the supporting stroma that I create. And that supporting stroma is basically a blood clot without blood. And so I supply the nutrients in a liquid media. And these are what the guys in my lab call my chia pets. And this is, in this case, a 2 millimeter in diameter piece of human placental vein. In America, if mom's okay, baby's okay, they throw the placentas in the garbage can. We have got a deal with the university that we're allowed to take them out of the garbage can and use them for research in an anonymous fashion. And these things are real blood vessels. You can see that they look like blood vessels. They anastomose with one another. And if you look at them at high power, they actually have lumens. And if you look at them, this is the basis of our newest patent. At the tip, where they begin to grow, these are called tip cells. They're solid cords of endothelial cells. And as they start to mature, they develop lipid or oil-like droplets in the center of the blood vessel. And then those oil droplets collect together, and that's what creates the lumen of the blood vessel. So you have lipids in here, fat, and surrounding them are the endothelial cells, and we've now discovered what this fat is an apolipoprotein, protein A1. And so one of the questions we ask is, are there unique genes that are never in a normal blood vessel that are expressed when blood vessels begin to grow? And that's really cool, because we can take the placental vein out of the placenta. It's not growing, it's just sitting there. It's just a plain old everyday vessel. and then we make these little chunks, and they begin to grow, so we can look at the genes that are in the normal blood vessel and in the blood vessel that now begins to grow. And so over here is the normal blood vessel, and there is no somatostatin receptor subtype 2. And if you look at the tissue-matched blood vessel, it develops somatostatin receptor subtype 2 as the blood vessel becomes angiogenic. That means it becomes now a target for all those things that people were talking about: radio-labeled somatostatin analogs, poison-labeled somatostatin analogs, etc. And this is one of our vessels, and you can't see anything because this is a control. But I'm going to show you that over here, or over here, this is where the vessel is, and this is the clot, and these are the new blood vessels growing out in here. And this is an antibody developed by Tom and Sue Odorisio against SST2, and a guy named Doug Balster, who was in their lab, did these stains. Here is the vessel disc in the bottom right. This is the cut edge of the vessel disc, and these are the angiogenic blood vessels growing out into the blood clot without blood. And as you can see, they all stain with somatostatin receptor subtype 2, but the vessel disc itself, the old stuff that isn't growing, that stuff down here, doesn't stain. What is really cool is the only time I've ever seen it is look how the nuclei of these things. So you see cytoplasmic staining, and you see the nucleus has somatostatin receptors in the nucleus. We then did what Dr. Shaw said to do. We took a human tumor, we put it in a mouse in his butt, and then we let it grow, and we gave the mouse a radioactive somatostatin analog. Now what you got to know is that the tumor we gave him doesn't have somatostatin receptors on the cells. So when you do the scan, how is the scan positive in the area of the tumor? And the answer is, it's in the blood vessels that feed the tumor, not in the tumor itself. So there are two ways to go after a tumor. Go after the SST2 on the cells of the cancer, or go after the SST2 on the blood vessels of the cancer. Well, the next set of problems that we faced were there were no human tumor-based angiogenesis assays. We developed this blood vessel assay. Could we do it on tumors? And yes, we can take a piece of your tumor and do the same thing. This is an example of when Phil Boudreau gets done operating on you, he will send me out a piece of your tumor, and we will chop it up and put it into culture, and we can do 60 replicates. That's 60 different experiments on each different chemical at each different concentration. And you will see we can do things like PTK, which is one of the Novartis drugs, Gleevec, which is a Novartis drug, Apothelone, which is a Novartis drug, RAD001. This is a drug from another company called Entremed, 2-methoxyestradiol. This is a common chemotherapy, Taxol, and this is something people use for gout called colchicine, and this is a biologic called Chinese sweet leaf tea. And as you can see, you can now pick which agent is most effective in this case. Not only have we got these vein-based assays, but we also now have a heart-based assay, so that, like, remember the work with Vioxx and Celebrex that took it off the market was because it blocked your heart's ability to repair itself by growing new blood vessels. We also have a skin-based assay for the dermatologist for psoriasis, an eye-based assay for the ophthalmologist, and a fat-based assay, believe it or not, don't look at me, that you can't get fat if you can't grow new blood vessels. If you don't have blood vessels to feed the fat, you can't get fat. So it's an idea for control of obesity. And so these people can now use these tests, like for Novartis, to screen their industrial pipeline. All the agents that you're about to see were kind of given to us by Novartis, and we took these and tried to create a clinical trial in a test tube. Could we take hundreds of people or 50 people, test them all the same way, and see what would happen? And what you see here are several lines. This is the effective RAD 001. The blue lines, the dark blue lines, are controls, and the light blue lines are treated cells. And you can see that in this particular case, this is not what appears to be a super potent compound. Whereas here's another one of their compound. These are the controls, and these are the tissue-matched treated wells. And you can see that now this PTK 787, which Dr. Odorizio has in clinical trial, is a very effective inhibitor of angiogenesis. And now this is the SOM 230, which is the universal receptor antagonist developed by Novartis. Again, not a very good effector of angiogenesis. Whereas this is Gleevec, a drug that was originally designed to look at and treat GIST tumors. And again, these are the controls, these are the treated wells, and you can see that the difference between these two are highly statistically significant. And finally, this is probably one of the things that's near and dear to our heart that we did at LSU as part of a clinical trial using a drug called apophyllone B. Again, part of the Novartis pipeline. These are like 26 patients that were taken to the OR. These are their untreated tissue. And this is their treated tissue. And you can see that a very effective anti-angiogenic agent. What else are we doing in the lab? Now we're trying to say, could we develop compounds that were non-toxic, that were plant-derived, and that you could use today as something that might be useful in the treatment of angiogenic-based diseases. And so this is an example here. Gallic acid is a food preservative used by Kraft Food in all their mayonnaise during the 1950s. This is Chinese sweet leaf tea. And we have other things like black raspberry that we're developing. And this is, again, the controls. And this is gallic acid. And this is the overall effect of the gallic acid compared to the untreated control. So a very interesting compound in the fact that it's already approved by the FDA as a food preservative. If you look at your can of Coca-Cola, it says benzoic acid. This is trihydroxybenzoic acid, its brother. And this is Chinese sweet leaf tea. It's a sweet tea that is grown in a very small area in China called Guangzhou Province. And the reason it's sweet is it contains a compound called rubicides. And you can see that an extract of that tea, which we now have treated several patients with, and these are about 30 patients whose tumors were tested. Again, these are the untreated, and these are the tissue-matched treated controls. So we have 4 natural products that we're trying to develop right now. One is the Chinese sweet leaf tea. One is a black raspberry product. One is a juice of a Polynesian plant called the Noni plant. And another one is a fig extract that we're starting some psoriasis trials at LSU. And this gives you an example that in a tumor, how we can test black raspberry. Here's black raspberry, and we keep adding more and more to the test tubes, and you can see that in a very nice dose response curve that the more you add to the tubes, the better your response. And here's, again, these black bars are the overall effect of the black raspberry. And here's a fig extract, and here again, the more fig you add, the less of the blood vessels that begin to grow. And here is an example of the Noni Juice. You can see here's the control growth over 14 days. Here is 2.5% Noni. Here's 5% Noni Juice. Here's 10% Noni Juice. And over, again, the growth of the blood vessels over time. But what's cool is if you let everything develop and you let the blood vessels grow and then start treating them, half you stop or you continue to not have treated, and the other half you start to treat, you can actually get developed blood vessels to start to regress. Well, one final thing before I go today, not all tumors in the same sites are the same, and that is really going to be critical because we heard about Ki-67 chromogranin, and the question is if you have a primary tumor, a lymph node tumor, and a tumor in your liver, are they sensitive to the same drugs, and do they behave biologically the same way? And the answer is, I think, no. This is an angiogenesis in the primary tumor, and this is how angiogenic its lymph node metastasis is. And this is the number of wells per 100 wells plated that actually begin to grow. So in this case, the mesenteric lymph node is much more of a threat because it grows so much more. And this is the actual growth of the carcinoid primary versus the lymph node. And this is the overall effect. The carcinoid primary has relatively less angiogenesis than its lymph node. So we continue to tinker, we continue to look, and I think that using human tumors from the OR, instead of throwing them into formaldehyde or freezing them where they're dead, use the living tissue as a miniature human being, as a miniature factory, to try to figure out what that tumor is doing. Thank you very much.
That was excellent, Dr. Woltering. We have time for 6 minutes of questions.
Shell?
This is a very nice model. You get endothelial cell to grow out, but do you also get parasites?
Yes. Actually, what Dr. Oberg is talking about is the first thing that happens is the solid part of the vessel are called tip cells. And then you develop this endothelial tube, and then after time passes, a cell called a parasite comes out and sort of covers up the tube, and that's part of the maturation product of this. Yes, indeed, parasites do happen. And I didn't show you that because that's on the other side. That's maybe 2.5, 3 weeks down the line. That's the last part of the maturation process. And once that happens, then the cells become much harder to treat.
Gene, I'd like to ask you, did you look, or were you able to look at MIV1 staining on the primary and the node in the one that grew the fastest?
Yes. Actually, we have a paper that's being done by one of the guys in my lab, John Lyons, and he's gone back and looked at Ki-67, chromogranin, synaptophysin in the primaries, lymph nodes, and organ metastasis, like liver metastasis, from the same individual taken out at the same time. And it's really very interesting that the Ki-67 is not necessarily the same in the primary, the lymph node, and the liver met. And it's not always the same way. In other words, some liver mets have the most proliferative indices. They're the most aggressive, whereas other things, the lymph nodes are the most aggressive. So it's not always the same same
direction. Does it correlate with your angiogenesis, though?
No, actually, Ki-67 does not, interestingly enough. And we stain all of our tumors for factor VIII. And we do a visual histologic angiogenic index. We count how many blood vessels per square millimeter of your tumor are in your tumor, literally by counting under a microscope. And usually that number is about 40 blood vessels per high-powered field. And so ours, we have things that 20% of the wells begin to grow, and in other tumors, 100% of the wells begin to grow. And they don't necessarily, what you see under the microscope is not necessarily what gets translated in real growth.
One point that I want to recognize Dr. Obert for, and it comes to my question, was several years ago, if I recall, they biopsied liver tumor lesions and then treated with interferon and re-biopsied them again. And I was at the Ludwig Institute presentation, and I was struck with how a treated liver lesion that was in situ and biopsied would replace themselves with a fibrovascular type of histology. My question, Dr. Woltering, and I recognize the difficulty in doing this, but have you ever been able to stabilize or see stabilization with one of your powerful in vitro drugs, such as CPTK, and then rebiopsy and put that tumor to see what angiogenesis does after treatment?
Yes, but not in a human. What we have done is done that, taken a human tumor out and tested it, put that human tumor in a mouse, treated the mouse, and then put that back into the culture system.
What happens?
And things are vastly different. And that's one of the reasons why we don't use the mouse model.
But I mean, the angiogenesis is, so you can't use mouse. The question is, does it slow the angiogenesis in a treated tumor?
No, actually it stimulates the angiogenesis.
In the mouse?
The angiogenesis in my assay, in the tumor that came from the mouse. It looks like the process of taking a tumor out, culturing it, and putting it in a mouse, and then taking it out again somehow stimulates the tumor.
We have a few more minutes, please, for questions or comments for all three people.
I mean, you have looked at somatostatin receptors, and we have also demonstrated that in human tumors and the newly formed blood vessels. But have you looked at other receptors, PDGF, alpha-beta receptors, and other tyrosine kinase receptors?
We now have 5 genes that are never in a normal blood vessel that are uniquely expressed in angiogenic vessels. One of those is there are 2 basic receptors for vascular endothelial growth factor, VEGF. And VEGF has R1 or FLT1 and R2 or KDR. And R2 is the one that is considered to be the important receptor for VEGF. And that R2 VEGF receptor is never in normal blood vessels, but is always in the tissue-matched angiogenic counterpart that we did. The same way with PDGF, or platelet-derived growth factor. PDGF receptor is never in normal blood vessels, but it's always in the angiogenic blood vessels. So there are 5 of those we now have.
Yeah, I'm a lay person, but before we all go out and start gorging on Noni Juice, blackberry powder, and all of that kind of stuff, can you explain that things that work in the lab, in the petri dishes and test tubes, don't necessarily work by ingesting stuff through the digestive tract?
Yeah, absolutely right. And anything that you saw up there, you notice that the fig is being used in a psoriasis skin cream trials, not orally absorbed. Noni is orally absorbed, black raspberry is orally absorbed, and Chinese sweet leaf tea is orally absorbed, and gallic acid is not orally absorbed. So those are all things that we have done, and before we use them in clinical trials in humans, we understand the degree, and one of the beautiful parts about it, having an assay like we have, is I can put you on black raspberry, I can wait for 3 weeks, I can draw a tube of your blood and use your blood to overlay that fibrin thrombin clot, that blood clot without blood, And if you have active degrees of drug in your blood, it will inhibit angiogenesis in my assay. So we can not only tell you what'll work, but then we can take your blood and we can take blood from somebody who's, let's say, on the RAD-001 trial or the PTK trial or SOM-230 and put it in our assay and get an idea of how it works.
And Dr. Gene, this is very exciting, but in follow-up, do you have to own a black raspberry farm to eat? And how many pounds is required a day?
And this is a good point. To give you an example, and the name of the farm is Stokes Berry Farm. It's an Ohio State farm where Dr. Odoricio came from and I came from. The powder is about $80 a pound, and the anti-angiogenic dose is about 1 gram per kilo. So like I'm 100 kilos, so 100 grams would be like 3 ounces a day. So it's a significant amount of, but then again, let's be honest. It's freeze-dried berries. I mean, it's not voodoo extracted with chloroform or something else. It's just a berry that was freeze-dried and ground up. So, I mean, that's non-toxic.
And it's available.
And it's available. And it's not, the FDA can't get on your case about it. Yeah, we figure that a black raspberry is 90% water. And so, like, you'd have to eat like a pound and a half of black raspberries a day. And the downside of black raspberries, interestingly enough, is if you've ever seen a side of beef, my father was a butcher. A side of beef, there's a stamp on it, USDA approved, and it's purple. That's black raspberry juice. It's the approved, FDA approved dye to put on there. So don't ever get black raspberries on your clothing because it will never come out.
Okay.
Thank you very much.
Thank you very much, and thank you for your attention.